Heyo, welcome to the world's best podcast. We got a pretty good guest today. We got a guest. His name is Wes Brown. This guy is probably the smartest human I've met in person. At least he used to be. I don't know what if that's changed. Okay, cool. I didn't know if it was sustainable. But area background, me and Jack met Wes in the fraternity. We went to Oregon State University, and I knew that you were studying to be a neurosurgeon, some sort of medical deal. And I go, this guy's pretty good. And here's I'll share my.
Oh, go ahead. We'll get into that later.
Yeah, that's what we're here to find out the updates. So my favorite Wes story is so you won this award for, I think you won this like 4 times in a row. It was basically just the best student in the, like, whatever college you were in, like the College of Medicine or something like that.
Agricultural Sciences at Oregon State.
Yeah, you're the College of Agricultural Sciences, and you won like best student. And then we found it downstairs in the kitchen, just like kind of under some like papers and stuff. And I just remember you being like, we're like, wait a minute, isn't this Wes's like award? And we brought it up to you, and you were like, oh, there it is. And then just set it on your stack of awards. You're like, I don't know, they just give me this shit. I don't know, I don't really care. It was like, this guy rocks. But yeah, dude.
Possessions.
Yeah.
They're overrated.
Yeah, we don't need these plaques. They cost $10.
Mountains.
Dude, I could see that working for you. Maybe be a Tibetan guy. Dude, all right. Well, I'm really curious. Where are you at now? Because you graduated Oregon State. You got your pre-med degree. And then I know that I talked to you a little bit before this, but basically you're still in school because it takes forever to get a PhD. But so give us the rough summary, the timeline from graduating college with your pre-med to where you're at now, where you're going.
Sure. So yeah, I graduated back in 2016 with the a bioresource research degree. So it's kind of like pre-med, but it's like research focused. So it's kind of an interesting kind of mishmash of stuff that you do in that degree. And then I applied for, first I applied for just medical school. And basically at the very last second, like a couple days before the deadline, I switched to MD PhD based programs because I liked research so much, and I kind of like my guidance counselor was like, why the hell are you like not applying for these things? So I applied for that, and then I went around and interviewed and stuff and got into the University of Pittsburgh. So that's where I am right now, doing the it's called the Medical Scientist Training Program. It's like your MD PhD based program that a lot of schools have, but it's like an 8-year program traditionally. So like they break med school in half, and so you take the 2 years of med school first and pass your like what's called the USMLE Step 1 exam, and then they throw you into graduate school for your PhD, and then you just do research for like 4 or 5 years, and then you jump back in to the second half of medical school, where you do all your clerkships and working in the hospital and like learning how to be an actual doctor.
Oh, that's like the residency.
It's not the residency yet, but it's like they throw you in the clinics because you don't know anything.
How old will you be when you get to go like practice as a doctor?
Well, once I'm a resident, I'll be like actually taking care of patients like a little bit more independently. That won't be until I am like completely graduated in like 2024. So yeah, 4 more years, or 3 more years. Oh my God, what year is it? So yeah, it's a long haul, but
that's a lot.
Yeah.
What kind of doctor are you gonna be, Wes?
I don't really know yet. Interested in internal medicine.
Okay.
And so probably going to specialize in something. Cardiology is like on my radar right now, but could be pulmonology, nephrology, GI. I don't know, something like that. Any of the organs, they're all.
You can go stick your fingers up everybody's butt.
Yeah, there we go.
That's the only reason I want to do it. Yeah.
I used to date a girl that was a GI or whose dad was a GI, and he'd always joke about how many fingers he'd stuck his finger, or how many butts he stuck his finger up in his life.
Yeah,
that's
testing you.
Jack, have your prostate checked recently? That's how he evaluates all of his daughter's boyfriends. He goes, I gotta see how your prostate. I gotta check your prostate.
It's a little inflamed. I don't know if this is gonna work.
They say your character is related to the size of your prostate. Man, man, that's a lot of, so how do you keep that? What keeps you going when you're in that many years of school? What keeps you patient and driven to just keep chugging?
It's a good question. Self-hatred? No. It's probably the fact that I like what I'm doing. Like, if I didn't
Mm-hmm.
like what I was doing, I probably wouldn't be doing it. Like, if I had
Mm-hmm.
to like really just like force myself to do everything and force myself to
Mm-hmm.
be awake every day, like I don't think it would work for 8 years
Mm-hmm.
straight. No, I like medical school. I loved it. Was really fun. I love learning. That's why like I'm okay with this much school. I like
Mm-hmm.
school. And so that makes it a little easier. And then research, I really enjoy research too. There's always like exciting stuff to do, stuff to learn. Most of the results you get are shit, but sometimes you get little like stones in the mud that, are
Mm-hmm.
little gold flecks in the mud that, get you excited about something and something that you can write up and publish. So
Yeah,
yeah, I just like it. That's why.
that's the best reason. That's
Yeah,
the best
that's
reason.
the
What's
only reason.
yeah. What's your research about?
So, it's an interesting like group. So we primarily do work in a field called chemical biology, and basically the idea is it's kind of like the inverse of biochemistry. And that biochemistry, you're like studying like cell processes and cellular reactions and stuff like that, and you have to memorize the Krebs cycle. Well, crap.
Mm-hmm.
We kind of basically take that, but we try to develop tools to better understand like cell processes. So the
Mm-hmm.
lab I work in is kind of a tool development lab. We like develop a lot of different techniques for
Mm-hmm.
trying to better understand biology. And so the stuff that like my projects focus on, it's a pretty broad group, so people work on all kinds of stuff. The stuff I work on tends to be something called genetic code expansion, which is a technique that's been developed over the last couple of decades. It's actually what I did in my undergrad too, with the
Mm-hmm.
MELD group over at Oregon State. But we basically incorporate unnatural amino acids, site specifically into proteins of interest, and use that to control the protein in some way to understand its function,
Mm-hmm.
and hopefully relate that back to disease.
How does that? I mean, obviously, you just blew my mind. I don't
Wes,
really.
you're doing
Genetic.
some cool stuff. Is
It's cool. It's fun. It's why I get up in the morning. Yeah.
So wait, so amino acids. Okay, you gotta be patient with somebody like me who and Reno who did not do a lot of the biology chemistry route. So amino acids, those are like I've always understood those as like the building blocks for life in general.
It's what
like
you need for your gains. Yeah.
for the game. Yeah, yeah.
Dude, hold on. Let's see
Tries,
this. Let's see the tries. Let's see the West tries
You
real
hitting
quick.
them?
Dude, we gotta see the
No,
West
no,
tries.
Wes Brown has the most legendary tries.
Oh!
Oh yeah. Um,
But yeah,
So-
they're building blocks of life. Yeah, proteins are made out of amino acids, and they're basically just strung together in beads on a chain kind of fashion. And they fold. The proteins fold into very specific, conformations that allow them to actually do stuff. Because just the string themselves doesn't mean anything, normally. So yeah.
what are the limitations of our current tools, and what are the new tools you're developing supposed to accomplish that the current ones cannot?
Right. So for studying like proteins in general and stuff, a lot of like especially earlier work and stuff, you take the protein, you express it in like bacteria, you make a bunch of it, and you can like study its function outside of cells. You lyse the bacteria, take it and like do some enzymatic assays with it, like oh it phosphorylates this protein, or oh it like leaves this protein up, that kind of stuff. Like basic kind of stuff that you can do outside of cells. What we want to do is kind of be able to take be able to control the enzymatic activity in a way where we could study it inside of cells, and do it in not a way that you have to kill the cells in order to do it. And so, for example, the stuff that I'm kind of focusing on is basically taking this ability to control enzymes, and studying zebrafish embryos and studying development. And so, for example, the main unnatural amino acids we incorporate are these photocaged amino acids. Basically, they resemble a normal amino acid like lysine, but then we stick a big mask on it so that it doesn't function inside the protein until we shine some light on it at a very specific wavelength. That mask pops off, and then the activity of the enzyme is restored. And this works super well in zebrafish embryos because they're completely transparent during the early stages of development. So we can take a laser and shine specific tissues and activate the protein, and down to single cell level with just really precise spatial and temporal control. So it allows us to like get like the most control that you can over like the protein's activity, with just great like characteristics of light. And so we kind of use that to study like kinases a lot of the time. For example, kinases are easy. Oh, sorry. This it's a protein that phosphorylates other proteins. It attaches a chemical group called the phosphate group onto other proteins, and that is basically a signal that's like it activates or can deactivate certain things within a cell, certain processes. And so we can take those and put them under optical control pretty readily and study their function during development.
Can I repeat back to you to see if I'm grasping this correctly? So basically you have some specific light wave that you're projecting into this cell, which is supposed to activate processes that you already know exist. These are processes you already know exist. You're just trying to like deliberately induce them at a specific time without killing the cell.
Right. Yeah. So they're proteins we know about. We know most of their pathways. There's a lot of stuff we don't know about these pathways. They can be interconnected in ways we don't expect. They can phosphorylate other things during different time points than maybe at later developmental time points. It's so context dependent. So there's a lot of stuff we don't know about how these pathways work. But yes, we can target a specific time point in development and understand better like how does this protein function in this very specific context, and how does that lead to say this developmental defect.
So, like in humans as well as fish?
I don't do any work in this stuff isn't being done in humans, mostly because that would be very ethically problem. Because basically what I do is I take zebrafish embryos and I inject them with all the mRNA and everything I need to do this. Let them grow up a little bit, shine them with light, to activate this protein, whatever protein I'm working on. And then look at development over time. Look at like, does it screw up the body axis? Do they like not grow a tail, or do they like not grow a head, or weird stuff like that? Or more specifically, looking at like heart development. Do they get like malfunctioned heart defects that cause like a lot of edema? Compromises their like circulation.
So is the ultimate goal of this research to figure out what mRNA, is the ultimate goal of this to find out how you eliminate defects or what causes defects in human children? Like how if someone has a
discovery based kind of thing. So we're not like using drugs or anything. We're not like trying to do drug screening to find something that like, cures disease right now. We're doing more of like the basic building blocks, trying to understand the disease itself. So like one disease that I'm trying to work on right now is rhasopathies. This is basically a collection of diseases that they have a lot of different phenotypes. That like heart defects is one of the most common traits that people with these diseases have, but they affect neurological development, craniofacial development. So we're basically studying these diseases because they all affect the same proteins. They all affect these signaling proteins, kinases that I just mentioned. And we're taking those mutants, we're photocaging them so they can't function, and then we activate them at time points so we can see like what stage of heart development is being impacted. How is it getting impacted?
Wow, this is good.
What other signaling pathways, are involved with this that this mutant might activate. stuff like that, and being able to do that in specific tissues, especially because this disease affects like everything. The RAS-MAPK pathway, which is where the RASopathies get their name, is like one of the biggest, most crucial signaling pathways in cells, like across like all eukaryotic cells, and so it affects everything. And so being able to specifically like pinpoint it to like heart cells versus like the whole embryo, it really helps kind of delineate like what is happening to what.
So how much is there a variation between embryos? Like are you using cloned embryos, or do you not need to control that hard for what's going on?
They're like wild type, as in they're still like they're the lab standard, I guess. They're like called AB star. Essentially, they don't have any transgenes present, stuff that we put in them to make them express, but they're still inbred in a way that they're all, pretty genetically identical to each other, so there isn't too much variation. This is true for all animal models, by the way, for mice, for worms, whatever people study, frogs. They're inbred enough so that they're all like genetically close enough to each other that you don't see the natural diversity that you would if you just went and picked up a frog out of your backyard or something. So they are kind of controlled in that way.
Have you guys heard about the Brett Weinstein? It might have been Brett or Eric Weinstein. How one of them did this research back in the 90s, and he discovered that like the lab mice had these like really abnormally long telomeres compared to regular mice, because they were selective breeding younger mice, and then like the telomeres were what were causing extra cancer in these lab mice. It's a wildly good podcast with Brett Weinstein and Eric Weinstein, and it's from a few months ago, or probably about a year ago. But it's just this whole story about how this is back in the 90s, where it became uncovered by Brett Weinstein that these lab mice were basically not good samples for all the tests they were doing on, and then like running human stuff on, and they thought it was like the cause of a lot of heart problems. And it's because they were selectively breeding younger mice and not letting them naturally get to old age. One of the consequences was like these telomeres at the end of like the cell regeneration were like abnormally long compared to normal mice, and they didn't also have this correlation between like cell reproduction and telomere length defined properly yet. So and then it like goes into how this all got covered up by the science community, and then like it got published like 5 or 10 years later. Anyway, I was just thinking about that because of the lab stuff, and I just wanted it. It was like one of my favorite podcasts I've ever listened to. I'll send it. I'll do a little post on it afterward. But it's watch out for that. You don't, you get all your from the same lab, and they're over controlled. They might be doing some selective breeding that could mess with your results.
Right, for sure. Yeah,
But I think they've fixed the problem since then. Or at least
yeah,
they have.
It's part of a wider problem of like scientific integrity and. A lot of it isn't down to like malicious intent, but there's just like nowadays a lot of people are realizing that results that get published by labs, strains that people are using of different cells or animals and stuff are not always reproducible, and that just leads to completely false information that wastes so much time and so much money, government money, because it sends people down these rabbit holes. These people get these like giant grants and stuff like that for millions of dollars to investigate something that might be based on a shaky premise.
Interesting.
Oh, really? Like that's a regular occurrence?
That's very regular. Yeah, like because that's one of the biggest problems in science. Something they hammered into our heads during my training and everything was about scientific integrity and making being able to reproduce your results, being able to being able to have like a general understanding of what's actually happening, what kind of experiments you're running and how you're running them, and able to be aware of any bias that might be occurring in your experiments too.
Which I feel like can be really difficult when you're talking about something as complex as what you're talking about. It's just like, how do you what order of magnitude size-wise are you dealing with when you're making these tools? Like how big are they? Like how big are these tools? How small are the measurements you're making? Like, because it feels like it's tiny, right? And so it's like, how are you making tools?
The measurements, like what kind of measurements I'm making for my experiments can vary from, enzymatic based assays and stuff like that, or fluorescent assays looking at like green or red protein production in embryos, or it could just be straight up phenotype of an embryo. So I like let them grow up to a certain point, compare them to their people I didn't treat, and just see like, oh, this one is significantly like shorter or messed up in some way that we were expecting. So that's the kind of
Oh,
measurements I
okay.
make.
Okay,
Yeah, the
gotcha.
stuff I make, fortunately, a lot of the experiments I do are pretty black and white. That's mostly because I'm in tool development, so I'm not going too much into like the nitty gritty biology, that where you do tend to see like very minor differences between,
Hmm.
test and control groups, and you have p-values to make sure like, oh, it's barely significant. We got a p-value of 0.05, like hooray, it's real kind of thing. When it still, like, that's a 5% chance of it like being a false, positive. So
Oh, okay.
that's when you get into murky waters, is when you're testing, yeah, things like that, and you're like, oh, this gene is like barely more upregulated than this gene when we added, this to the water or whatever. So like that must mean this, and it's like, well, it's like when you have a tiny difference like that, and it's barely significant, it's like, does it really mean anything? You have to really be
So how do you
suspicious.
so how do you control that? You said, yeah, how do you maintain that integrity? Because you talked about that where they like really hammered it into you, like, no, this science, it's got to be repeatable. You got to have integrity with this. You don't want to waste grants on something that's a total rabbit hole. How do you maintain that integrity? Like, is there, what's the process? Is it just being honest, or is it like you have to do a different
It's
process?
it's being rigorous. That's like the name of the game. It's being, writing down like everything that you do, a protocol that you have that you use a bunch, like be as specific as possible. Like, what are you
Hmm.
doing? What's the temperature you're doing it at? Like, just every detail you can think of, like, make sure to include it, because sometimes tiny details make the difference. They make something work or not, and
Hmm.
you don't even realize it. Um,
Hmm.
just the way, that like some of the bigger journals like Nature and stuff like that, they're having you like, include very detailed protocols, making sure your antibodies that you bought from some outside source actually function. um,
Hmm.
Stuff like that is becoming a lot more commonplace. More and more journals are like kind of signing up onto this rigorous assessment of, the science that you conducted. Because before, like, if you ever read like some of these papers from like the 1970s or like 50s or something, like people were just kind of publishing like whatever they wanted. Like, you could do something in your basement and like go publish it. There was just
Really?
no, yeah, there was not a lot of like rigor that was included with like really really old publication stuff, because it was just like, you give it to us, we'll publish it kind of thing. But now that not only is like all research, most of the research in this country being publicly funded taxpayer dollars,
Hmm.
so and there's just so much more of it going on right now, and people base
Hmm.
their careers off of it. There has to be some kind of holding to account for people to make sure that
Hmm.
they like are, you know, not wasting all of that taxpayer money
Hmm.
or other people's time.
Yeah, on on something that just happened to be a happenstance of a freak genetic code or something, like some genetic alteration that they didn't see. Is that what you mean? So basically, they
That's
weren't
one
rigorous
great
enough.
example, because with CRISPR-Cas9 stuff going on right now, the CRISPR-Cas9 complex like will
Do you
sometimes
want to? You
randomly
should
induce
explain to
genetic
all of our
errors,
viewers what that is.
instead of your target gene. And so you have to, like, when you publish something and
Wait, Wes, I'm going to pause you. You
oh
got
yeah,
to. I need to know what CRISPR-Cas9 is.
oh yes,
I need to know what that
CRISPR
is.
CRISPR-Cas9, all the rage. It's gene editing. So
Oh, okay.
basically, it's a protein complex. So it's Cas9 is the protein. CRISPR is just like the name of like the whole system. It's like clustered, regular, interspersed something palindromes. Re piece. I don't know. It's basically the idea is you have this Cas9 protein. It hooks up with RNA that targets it to a specific piece of the genome to cut it. And that's all it does. It goes in, cuts the DNA at that specific site, and then you wait for the cell repair machinery to get there and repair it funky. So it either repairs it and induces like some mutations and crap, so it basically destroys that gene that you targeted, or if you include like another piece of DNA in there that's supposed to help essentially repair that break, but repair it in a way that you want it to be repaired, so you can insert some kind of like new protein or something like that into a system. That's what it's for. And so there's a lot of it. People are using this all over now. It's like, super
Like
super
the new
popular.
vaccine, is that CRISPR-Cas9 how they did it?
No.
They're making us more compliant. They're forcing government compliance.
Yes, they're implanting Bill Gates.
What's the deal? Is that
microchips
safe,
into our
Wes? Can we all
DNA.
take the mRNA vaccine? Is that safe?
I took it. Yeah, I got it. And
Yeah,
it's great.
but it works.
Yeah,
No
Bill Gates
coronavirus
is
sense.
telling me to say that. Yeah.
Yeah,
He's standing
you're
right
chipped
out to me
now.
right
You
now.
might be chipped now. How do we feel when you
I'm
track?
transmitting 5G. I'm a 5G tower.
Yeah.
You're a 5G node.
Us all
That
in
might be a
5G
bonus.
towers.
If that was the side effect, I'm in. We need more 5G towers.
I
That's
am
how we
showing
work.
you that was actually like my mom sent me this link and she's like, oh my God, did you see this? And it's like this woman, she's like, I'm an OB-GYN at this hospital, blah, 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 all her like records and stuff. And then she says, if you take this vaccine, the metals in the vaccine will turn you into a radio tower for 5G. And I was like, okay, like that's enough.
But
Like
somebody with credentials said this?
someone supposedly, I don't know if they actually had credentials. They just went on there and said what they wanted to say. So maybe they did, but maybe they did. There's
They coulda
plenty of
lied about their
doctors who
history.
do stupid shit, So it's like it happens, but it's
So you're
really-
telling me if I get the vaccine, I'll have better cell reception.
you'll have wherever
Dude.
you go. You
Yeah,
go inside
because
a
I'm bringing it
cave,
with
you'll
me.
have it.
Yeah.
They should have started with that. That's how
Yeah.
they should sell the vaccine. I mean,
That would've
listen,
been-
secondarily, it'll solve your COVID problem. But primarily, your cell reception will be outstanding.
Yeah.
I would take that. Imagine going into AT&T, they're like, oh, do you want to upgrade to 5G? You go, yeah. And they just stab something in your arm.
Do you actually know the function? Like, because they keep calling this like the mRNA vaccine and all that. What does that mean, and why
I'll
is it
tell you
different
all
than
the
other
deets.
different
Woo.
vaccines?
I
I'm
think
really
it's
curious
one of the most
about this.
exciting things in science right now.
Yeah,
Oh,
I want to
boom.
know. This is good.
So basically, yeah, mRNA vaccine. It's the mRNA. So what they did was previous vaccines, they used to just take a virus, like kill it or affect it in some way so it couldn't infect you as well, and then inject that straight up into you, and then your immune system takes over. For the mRNA vaccine, we know the sequence of the proteins on the surface of COVID. We know we can take that sequence, make mRNA, and inject that.
What is mRNA?
Okay, so mRNA is it's called messenger RNA. That's what the M stands for.
Mm-hmm.
It encodes a protein. So basically, once that gets into a cell's cytoplasm, the ribosome will hook up to it and start making protein off of it.
Mm-hmm.
And so basically, what we're doing is telling your muscle cells, your own muscle cells, to express this viral protein. And that viral protein is what the immune system recognizes in order to be able to create antibodies against COVID. And so how it works is that, so it sounds so simple, but people have been working on this thing for decades. But they have to package the mRNA in basically a liposome, which is something that it's like this kind of fatty like layer, kind of like a cell, like has a lipid like layer. They basically coat it in this kind of fatty layer that allows it to pass into your muscle cells where they inject it, like at the site of injection. And once it gets into the muscle cells, the mRNA is released. It makes the protein. Then your cell has a way of, its natural immune system is able to take proteins that it doesn't really recognize, and it chops them up into pieces basically and presents them on the surface of the cell. And it's basically like a little flag that's saying, hey, immune system, I got something weird here. Can you come check it out? Is this bad? And then that's when your T cells come over and are like, huh, this is your own proteins. What the hell's wrong with you? Like, calm
Mm-hmm.
down. Or like, this is foreign. What the fuck? And then it
Mm-hmm.
like activates the immune system. It'll go activate B cells and make antibodies. You get the whole activation cascade of the immune system. So basically, we're getting your muscle cells to produce the viral protein, present it to T cells, and activate the immune system.
Okay.
So so why can't we just do the dead COVID cells?
You
Why can't we
could,
use that old method?
and I think one of the vaccines is based off of that. One of them, like in China or something like that.
No, we don't want that one.
But I mean, yeah, like that's the go-to. That's like what we have, used for a lot of our vaccine. Most of our vaccines is like live attenuated or dead virus vaccines.
Mm-hmm.
The problem is it just like sometimes doesn't work. Like it depends on the virus.
Yeah.
It differs so much between virus to virus or bacteria to bacteria.
Okay, wait. Let me see if I conceptually understand this mRNA thing. So it's basically you send a package of code to the muscle cells,
Yep.
and the muscle cells run this code, which produces these foreign proteins that are basically identical in nature to the proteins that the coronavirus has.
Yep.
And then so your muscle cells basically is like doing the creation of the virus,
Yeah,
and it goes,
protein production
and then
factory.
and then it goes, I don't know what to do with this. Boom, sends it out into the ecosystem of your body, and then the ecosystem of your body comes in and says, oh shit, we got to do something to protect against this, and now you have immunity.
So it's not exactly like shooting it out into your system. It's like the T cells like run around your cells and they're checking on things, making sure everything's okay. And so the muscle cells that made this protein and are presenting it on the surface tell the T cells basically the T cells recognize that as foreign, and that'll activate the immune system.
Wow.
And then everything happens.
Seems like the coolest technology. Why? Like, this is
It's really cool.
way more
because-
interesting than I thought it was.
Yeah, it's like it sounds so simple. It's like, oh, just make the protein like in your body. It's like so much more difficult than that because mRNA, first of all, is like super unstable. It degrades like if you look at it funny. So that's why like you hear about the storage and stuff. They're like, oh, this one's cool because you can store it at like in a refrigerator for a week and it won't degrade. It's like okay. So it's like cheese or something. But that's great because normally it's not that stable. What they had to do to the mRNA to make it a little bit more stable is add some modifications to it so that it doesn't just get chewed up as soon as you inject it. And, to make it
And that's
more
part of that
shelf-stable.
fatty layer on the outside. Is that helps like make
That's
it
more for
not break up
transmission.
into
Yeah, that's to help it get into cells because mRNA itself is a very charged molecule, and charged molecules don't go through cells normally unless there's a specific transporter for it, which there isn't. So you have to coat it in a kind of a fatty bubble to get it into cells.
Like when you give your dog medicine and you put it in a little bit of butter
A
or
little
peanut butter.
Yeah.
Yeah.
Exactly,
Okay.
yeah.
So we cover the mRNA with the peanut butter, and our muscles like, oh, dope. Some peanut butter.
That's
Okay.
what they use actually is peanut butter. So if you have a nut allergy,
See,
you can't get the vaccine.
it's not that complicated. They just took some protein, they put it in some peanut butter, and they put it in. Yeah, that doesn't. See, we're getting it.
Yeah.
So it feels like the tools you're developing would help to further this kind of technology. Is that right? Or no?
Maybe it's hard to say. Like right now, it's limited because the proteins that you produce, that's great for vaccines. It's like a perfect storm for vaccines because you can produce the protein in a specific site, and then that gets presented. It gets chewed up. It gets presented, and then the immune system mounts its response. But you couldn't use it to produce like exogenous proteins really well.
What's that? What's an
because-
exogenous protein?
Sorry, like proteins that your cells normally don't express. So
Oh, okay.
you can't really use it to do that because, like I said, it gets chewed up, and then it gets presented. So that's part of the problem. And also, you're only expressing the protein in like muscle cells that got directly like injected into when they jabbed it into your arm. The rest of it, gets kind of interspersed. It might get into the bloodstream, but and what eventually happens is it like just goes into the liver or the spleen and gets like degraded there. It doesn't really go anywhere else. So that's why it's kind of like you can't like express a protein in any like tissue that you want, this kind of system. It's more just like where you inject it, it'll get expressed there for a while, and then it'll
But
like
as long as it's expressed somewhere, your body develops the antibodies.
because it's for a vaccine, it doesn't matter. Yeah, get
Yeah.
it. The muscle, it makes it so easy because that's where you inject things anyway. Just inject it there in the muscles. The muscle will produce the protein for you, and then that's what you need. But if you wanted to say like create a protein, like gene therapy, what gene therapy is you try to produce a protein that maybe your disease cells don't produce. You can't use this method because it's not going to really get you the tissue you want. Like if you're trying to treat cystic fibrosis, and you just have to express the correct protein that people with cystic fibrosis don't express, you couldn't really do that because the mRNA is not going to get to the lung cells that need to produce the protein. So it's got its limitations there. But for mRNA, for vaccines, this is like super cool because it's easy as hell to do. Like I could make the mRNA myself. Like if I had the code for
Is that what you did for your vaccine? Did
I
you
could just go
homebrew
make
one?
it in lab right now. Like anybody can do it. Everyone has like a mRNA transcription kit in their like freezer somewhere. So you could transcribe the mRNA for whatever disease, like whatever virus or bacteria or whatever, to produce the protein, the surface protein that the antibodies would bind to, and then inject it, and then you should be able to get an immune response, hopefully, from it. That's the hopeful, that's like the very optimistic view. But then, like you mentioned earlier, why don't we just do live attenuated virus vaccines or dead virus vaccines all the time? It's because they don't work, and the mRNA vaccine might not work for some viruses. For COVID, it obviously does, but for some viruses, maybe it won't. And that's because the immune system has to get sufficiently activated in order to actually mount a response. And why the dead virus vaccine doesn't work is sometimes it just doesn't, your immune system doesn't get that activated. They're not that, triggered by it. They're just like, oh, okay, whatever. And
They're
then
not triggered. They don't care.
Yeah, not triggered. Yeah. And so,
So
immunology is like ridiculously complex, and like we're learning more about it like every day. But there's just so much that goes on in activating your T and B cells, that we're still like trying to learn about, and that's kind of the basic underpinnings of like how vaccines work, is activating those cells.
So so instead of trying to get your T cells to react to something, is there any research on just training your T cell itself, where you can just basically put in some T cells, like trainer T cells, and they come in, they're like, hey guys, so we know how to handle this virus and we're going to teach you.
That's a great idea, because that's something that they're doing actually already. For cancer immunotherapy, they're called CAR T cells, chimeric antigen receptor T cells. And basically what you do is you take the T cells out of somebody, their own T cells, so that they won't react to like other people's T cells. This makes it so there won't be some crazy reaction when you inject it back into them. You take their own T cells, you get them to express basically an antibody, a receptor that should recognize some kind of surface protein on the cancer that's overexpressed, something that's specific to the cancer. And then you re-inject T cells into that person. They go and attack the cancer and kill the cancer off. And that's work. It's really another really exciting field of science that they're trying to work on. There's of course always like hiccups and stuff to it. They're still trying to get it to it. I think it works great on like leukemias and blood cancers, but on solid tumors it's a little harder, because you also have with solid tumors, it creates its own environment, its own microenvironment, and where it's immunosuppressive. And so they like are producing, these signals and stuff that are inhibiting T cells from doing their job. And somehow, these cancer cells have like developed that kind of like invisibility cloak to the immune system. Because normally your immune system does attack cancer cells and gets rid of them. It recognizes bad cells, but those bad cells have to develop a way of getting around your immune system in order to thrive and become cancer.
Interesting. See, here I was thinking cancer was easy to solve. We were just lazy about it.
Right,
But it turns out it's actually pretty tough.
It's actually
Kind of
kinda
complicated. Yeah.
That's crazy that cancer, that the tumorous ones, like the physical tumorous ones, that they hide. If
Right.
they actually hide from your immune system. Is there any reason, do we know why it does that? Is there any sort of like historical reason? I mean, is there any guess as to why that is or what it's
I
doing?
mean, why is always a weird question because like so many things go wrong in our bodies, and it's just weird. Like cancer, like why are we so susceptible to it? I don't know if it's just because there's so much that could go wrong. It's amazing it doesn't always go wrong. That's the flip side.
Yeah.
So it's amazing that, everyone doesn't get cancer by the time they're 20. Like it's, there's just so much going on, so many checks and balances that your body does all the time, that sometimes it slips up and, misses one thing and then you're fucked. So yeah, the tumor microenvironment, that's like an active field of research. Like that people are really interested in that, trying to understand like what specifically is allowing these tumors to, evade host immune systems and stuff like that. There's other weird things like they could be more acidic environments that can screw up, the T cell response, perhaps, and innate immune system responses. There's all kinds of weird things that can happen in a tumor microenvironment.
Yeah. So these T cells, is it an issue that you can't? So you have to take someone's own T cells and then train those because you couldn't like because a T cell is just a cell, it's just a genetic code, right? So theoretically you could clone it, I'm assuming, or is that not possible?
I mean, yeah, T cells, you could, clone T cells. And I think what they do here is they basically like transfect the T cells, as in they basically get foreign like RNA or DNA into T cells to produce the receptor that recognizes whatever tumor cell protein they're targeting. So that's how they do that.
Okay, so you couldn't like manufacture a super T cell, just like a Terminator of T cells, and put it. It wouldn't be able to go into your body. Like your body would kill it. Like
if you like produced a T cell that is like not seen as self, then your immune system would kill it probably. Yeah. It wouldn't allow it to survive. Our immune system can be very finicky. Yeah, you don't want to inject foreign. That's why blood typing and everything's so important. You can't be like it's more than just like A or B or AB or something like that. Like there's more stuff that goes on to make sure that the blood that we're giving you is like a perfect match. Otherwise, your immune system will go on the fritz and kill you. Same with like organ transplants, like that. Having to find the match. It's like it has to be perfect match, or you will die. Or the organ will just get like destroyed as soon as you So,
and even if it's like that close to a perfect match, you got to take immunosuppressants for your whole life
right.
too, don't you?
Right,
you get
exactly.
Organ transplant.
Yeah, you do. So it's just your body does not do well with foreign stuff being put in it.
It's crazy that the organ transplants work at all. Like that you can just suppress the immune system, right? Calm down, body. This is the only reason you're still alive. I know it looks like a foreign object, and it's performing a function, so just chill out. You're going to want to attack it for the rest of the time it exists, but just stop. It blows my mind that we were able to just put one organ in somebody else and just perform the function.
Yeah, it is.
They what do you know anything about the research that they're of cloning someone's organs? Where you can take, I mean, I've read just tiny, very high level articles about it. I thought it was cool, and so I'm curious if you know more about it. Where they'll either 3D print an organ, like they'll take some good cells off someone's like bad bladder or whatever it is, and then they'll replicate it, 3D print it, and then put it back in, and then there's no immuno response because it's the exact identical bladder. I know they're trying to do that with kidneys or livers, but obviously the more complicated the organ, the more complicated that function is.
Right, liver's easy because it's literally like a mass of cells with some architecture, but it's not as complicated as, say, a heart. something like
Here's
that.
a cool fact about livers, which I'm sure Wes knows, but Reno, you might not. You can do live liver transplants. So like I, a living human, could give half of my liver to somebody else and it would grow into 2 different livers.
Yep.
Wild.
Isn't that crazy? You just cut away half and it's so regenerative. It just, both halves just rebuild into a whole.
Mm-hmm. Yeah, it's so
Interesting.
amazing that people are like trying to like study like why can the liver do this? Why like wouldn't it be great if the heart could do that or something?
Yeah.
Yeah, for sure.
So is there research on that? The 3D printing or the cloning of organs?
Yeah.
Like with the liver.
To
So
any question that starts with "Is there research on that?" is yes.
yeah. 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 That's a good point.
For sure. Like, yeah, organoids, for example, that's kind of what the first thing that pops into my mind when you talk about like taking cells and like trying to grow another like organ out of it. It's like in the field of bioengineering, so I'm definitely like not super familiar with it, but basically the idea is you have to take some cells, put them in a matrix that the cells are happy in, so that a lot of like weird hydrogels or like different matrices that simulate like the extracellular environment of your organs and stuff, so that they have to have the right mixture of growth factors, the right mixture of like pH and salinity and like all the other like small molecules that you need to make sure that this thing keeps growing and doesn't just die. Because but except for cancer cells, your cells, when they're taken out of where they're used to being, don't want to live anymore. They die very quickly. And so you have to make sure like everything's perfect. So that they basically take those cells, put it into like a cozy like environment that's like perfectly suited for those cells to help them kind of grow and proliferate and sometimes differentiate into like the right cell type so that they're actually functional. Because also part of the problem is all of your cells, most of your cells are not stem cells. They're terminally differentiated, which means they can't really grow anymore. They have a very limited capacity of being able to proliferate, because like you kind of mentioned telomeres a little back at the beginning of the podcast. Well, those telomeres are set in stone when you're a differentiated cell. They won't so each time you replicate, telomeres get cut off little by little by little until it starts cutting into the important stuff that you don't want to get cut. And so the thing about stem cells is that doesn't happen. The stem cells can keep their telomeres long with special enzymes that allow them to replicate the telomere itself. And so that's part of why you have to take stem cells to do something like what you're talking about and to grow organs out of. And you have to differentiate those stem cells into the proper cell type. And let them keep growing, but not differentiate too soon, because you want them to keep growing to be, a large enough mass before they like are terminal.
Oh, so you want the stem cells to just keep growing and growing, and then they start to decide what they are once there is enough of them.
Yeah, because it's all very orchestrated. You have to, start from a progenitor cell or a stem cell. And it has to grow a bunch, and then something has to trigger it to start to differentiate once it gets to the right size, the right number of cells. And then the differentiation is what gives it its unique attributes, like being able to pump, or, produce special proteins and excrete them into your stomach or whatever. Stuff like that. So, it's complicated.
That's super complicated.
I can't believe anybody knows this at all. Somebody went and figured this out. This blows my mind.
What are your thoughts on? I'm going to ask questions that sound dumb, obviously, because I know nothing about it. But stem cells, I feel like. Oh, here's my history of stem cells, but I'm really interested in them. Is I know at some point everyone's flipping out because they only came from fetuses or something. So everyone goes, "We can't get the stem cells from the fetuses," and then they made it illegal. And then they stopped research in the United States. But then I'll go on like Joe Rogan. They'll talk about like he went to Mexico or like Russia or something for stem cells for his knee. And that sounds terrible. But then he says, it regenerated. I don't know, some part of his knee. Like the stem cell research actually is pretty useful, but we won't allow research in the United States.
Right.
It's like that defined for me. Even what is a stem cell? I don't know. What's
stem cell is a cell that is pre-differentiated basically it means it can continue replicating and eventually differentiate into the cell type that you want it to turn into. So
it's like a building block
embryos,
to be any cell.
it could. So yeah, basically, it's a little more complicated because it's like a hierarchy. So like at the very top, like when you were one cell old, that could differentiate into anything. But then as you go down, you turn into 1,000 cells, millions of cells and stuff. Those cells start to get categorized into different kinds of progenitor cells that, they can't turn into everything, but they can say turn into heart or vasculature or, something else. Or, these can all turn into endothelial cells, versus these can all turn into like, epithelial cells or something like that. So yeah.
So the closer you get to the original cell, the more options you have from this particular one.
Right.
Okay, then what are they? So like, then back to Reno's question. What's like Joe Rogan doing with his knees? Like, how's that helping out?
So it's probably not embryo. So it's probably not embryonic stem cells. Those embryonic stem cells are like the very pre-differentiated cells. So that's why that has come up. They're like, oh, it's these cells you can differentiate into almost anything, because they just haven't. It's such a young embryo before it's developed its whole body yet. But there's a lot of other options for getting stem cells that don't require embryos, and that's something that I don't think is as well known and should be more well known. And there's a lot of active study in the U.S. in fact going on as well. Like for example, one big one that's pretty exciting is induced pluripotent stem cells, iPSCs. This is basically taking your own differentiated cells and treating them with some chemicals to essentially reverse time. I don't know. I'm not exactly sure how it works, but essentially.
Oh, here we go.
Into stem cells. And so that it basically lets them go take a step back, and then they can re-differentiate it into a different category of whatever you want. And so perhaps I don't know what Joe Rogan was doing or anything, but it could have been something like that. You get stem cells from your bone marrow contains stem cells. That's why there's bone. Oh gosh, my cats are going crazy. But your bone marrow contains a bunch of stem cells. That's why there's bone marrow transplant. They're stem cells, but they can only really differentiate into other types of blood cells. White blood cells, platelets, red blood cells, that kind of stuff. But yeah. So it doesn't have to be coming from embryos. That's kind of like what everyone thinks of when you say stem cells, but it's actually a lot more. There's a lot of other sources too. So, but
it's basically just when you deliberately cause cells to differentiate in the way you want them to help aid existing conditions.
Right. That's
Is
what
that
you
a
try.
fair definition?
They're a pre-differentiated cell that you hope. I'm not entirely sure how when it comes to actual human therapy. I've heard a lot of not so great stuff in the news about like certain stem cell therapies and how they aren't very well characterized, and there's not a lot of clinical trials that have like shown their efficacy yet, and that's probably why they're not as like popular in the media right now, and not everyone's talking about it because I think there's still a lot of active research going on to make sure that they work. But the idea is I think you are able to insert these stem cells into the environment where they'll hopefully differentiate into the correct cell type, or they differentiate them and then inject them. I'm not entirely sure, but there's so many things, there's so many complications that come with that, because not only do you have to make sure they're differentiated into the right cell, you have to make sure that when you inject them into wherever site, that they actually stick to that site. They don't just float away and die. That they decide to stay there and keep living. That's a hard thing to do with cells, especially like these things that haven't been growing there. You grow them in a dish or something, and then you're trying to tell them, hey, stay put, grow, just live here. When it's very easy and then immune responses too. That's another thing. Like, if your immune system doesn't like it, then it's going to die.
Yeah, that's a really good point, is that if you put in the foreign stem cells, then your immune system would just kill it immediately. They go, this is a foreign protein. I'm definitely going to kill this thing. This is what all I do. As your T cells, all I do is I kill foreign stuff, and that's a foreign thing.
Yeah.
So can you hide it? from your immune system?
So I'd assume for like the induced pluripotent stem cell stuff, like they take that from your own cells. I assume they could take that from your own cells, pre-differentiate it and redifferentiate it into whatever they want. And that probably won't have an immune response because it's their own cells. Just like with the CAR T stuff, they take their own T cells so that they don't have an immune response. It's kind of, they take someone's own cells to make the stem cell. But yeah, if you're going to inject them with some other person's stem cells or something, it'd probably not be good.
Yeah.
And have a bad time.
Yeah, you could have a bad time, bad trip. Interesting.
Well, Wes, how much time did you schedule for this? We're butting up on an hour. I don't want to keep you all day.
However long you need. I'm going to go do some work later today, but other than that, I got a free Saturday, so
yeah.
Whatever you guys want to do.
What's the most stepping outside of the like let's get a little step outside of the exactly what research exists. What's the most exciting thing right now for you right now medically? Maybe we already talked about it, but in terms of medical technology, what do you think is the most exciting that you read about? You go, this is badass.
Right. Yeah. I kind of mentioned before, the mRNA vaccine stuff. That's like very hopeful. I feel like they could, and they're trying to like use it to treat a bunch of other viruses and stuff that we don't have vaccines for. So that's really exciting. And then of course gene editing and stuff like that is always, I think, really exciting. It'll be cool to see where that goes, and if they're able to clear a lot of the hurdles that are in the way currently.
What's your philosophy on that, on gene editing, like designer babies and stuff?
Oh, well, yeah, designer baby, no. Using it to treat disease, yes.
Why? What's the difference? I don't know what the difference. To me, it just seems like there's your gene editing. What's the difference between doing it one way or the other? Can you break that down for us? Because
I mean, this is kind of my own ethics, but a lot of it. So, medicine is supposed to be for helping the sick and like the diseased and like the downtrodden, but if you start bringing like designer gene editing like stuff into it, it's not medicine anymore. And when you're taking something that is so rooted in science and medicine to do something that would only be available to the most elite, richest people in the world, it not only creates like a massive health disparity that I could see creating a lot of dystopian future novels, but it just like completely like corrupts like the point of medicine.
But it's possible. Like these are real.
Yeah,
I feel like it's possible.
Like if we
how do you prevent society from doing it
or something like that? Like we could like do all kinds of crazy off the wall mad scientist stuff and try screwing around. We take embryos, like I inject zebrafish embryos and get them to express all these proteins with pretty good ease. You could do that with human embryos too, and try to it'd be very easy to genetically modify a human at that stage, just because they're one cell. You want to knock out a specific gene or insert a specific gene or something. That's relatively easy to do with our like model organisms. The difficult thing to do is using it to treat like fully like developed humans, because we're made of like trillions and trillions of cells, and you have to try to modify all the right ones. So yeah, it's stuff that's definitely possible. It's just not ethically sound.
Right. Like, could you just, whoever has the most money could just have their kids be the smartest, most athletic.
I mean,
just biggest kids.
Turn into something like that. They're like knocking out genes here and there. And let's face it, we're as humans, as a race, we're not that smart. We there's going to be so many unintended consequences that come from something like that. We're taking millions of years of evolution, and crafting our genetic code to be a specific way, and it's obviously not perfect. It's not like well-engineered machine or anything, but we're doing something. We're like, oh, knocking out this gene should do this, and we have no idea if there's other unintended consequences that come down the line, like making
secondary and tertiary
to
effects.
certain diseases or to cancers or to anything. And the thing with CRISPR-Cas9 is it's germline transmissible. So if you were to edit your germline cells, it'll get passed down for every generation after you. And so like you don't know what would happen.
Oh damn!
Wait,
So
like you
it's
pass
not
it on
just
to your children.
If it affects the germline cells, yes, the cells in your sperm, yeah, it'd have to affect the sperm making cells.
We wouldn't just be affecting the person; we'd be affecting every person that ever comes from that original person.
You're taking control of evolution.
Right. Oh, for sure. Yeah, it would. You could like do that.
What the?
Yeah.
So it could get real fucked. And there's just. We don't know. It's first of all, it's not a perfect tool. Like I said, already lots of unintended mutations and stuff come from using it. People are trying to hammer that out to make it more efficient and not have so much background mutations. Also, like if we're just knocking out stuff because we're like, oh, this will like make us stronger, or this will like make us more resistant to this disease or something like that. And they're just using that to like treat like any Joe Schmo on the street. Like you don't know the benefits aren't going to outweigh like what are the risks that could come from this. Like we don't know if this has some other impact, that could make us more susceptible to some other disease or like royally screw up, development or something like that. Like it's hard. It's that's why I don't know the exact rules, but I know when CRISPR was blowing up, a lot of like world-renowned scientists got together and like were like, okay, these are the rules of conduct. This is what we can do and what we shouldn't be doing with this technology. And part of that technology is not affecting germline cells. We should not be targeting germline cells to do this because it could have there's unintended consequences that could get passed on for generations of generations. And so that's part of it. And then also the second part of that was like not using it to treat disease, not to, impart any like, special attributes and stuff like that are not, related to disease.
Right.
Okay, so at its current state, how close are we to be able to be like, if somebody wanted to do it? I know I agree with that it's unethical, but I am just curious because if the technology exists, it feels like somebody's going to do it. How far close are we to being like, okay, I want my kid to be 6'4", blue eyes, 9-inch cock, and chiseled abs? Like, is that so that's not even in the realm of
blue
what we're
eyes
capable
maybe,
of doing.
So for a lot of stuff, it's like most things are not single genes, right? So height is not something related to a single gene. Cas9 is good for targeting single genes to induce mutations and stuff. So I don't even think we understand like all the genes that are related to like height and stuff like that. I think for like eye color, hair color and stuff, that might be more simple. Melanin and production and stuff like that. But yeah, that the technology is still in development. Like it's still getting the trying to make it more efficient, trying to make it work. That's still going on. And then also just understanding genetics in general and its relation to development is overall.
So we're not really at an immediate risk of like Russia or China developing this race of superhumans that comes over and takes over us. That's not. We're not quite there yet.
There's some stuff like if you read around, like I remember seeing in the news, some like backyard biohacker people or something like
backyard biohacker.
Cas9. Yeah, that's what they call themselves. And they take pride in freaking idiots.
There's someone you can buy them online, CRISPR kits, right?
Yeah,
there's like guys that'll show you videos. They go, yeah, you mix these things, this will make you healthier, and they just inject themselves with CRISPR, and they have no idea what they're doing.
It's so stupid.
You can buy these for like $200. It's not even expensive.
It's
CRISPR kits.
Backyard biohacker. That's a cool name. That is a cool name. They did brand themselves well. I gotta give them credit for there.
Right. They make these kits. Like one of the genes that a lot of these people try to target is myostatin. which is like a known gene that like inhibits muscle growth. Probably because like when we were Neanderthals and like barely getting enough food to scrape by, we didn't want like uncontrolled muscle growth to like consume all of our calories in a day. And so like, you know, this gene exists, and so there's like these biohacker people are like, oh, I'm going to get like bigger biceps and inject myself with Cas9 to knock out myostatin and stuff like that. Of course, it never works because they're stupid. It's like it's a bad premise already, and it's like you shouldn't be. They're injecting themselves. Like, kind of, so I mentioned like with the mRNA stuff, you have to like have a lot of things working for you to actually make it work. You can't just inject mRNA and then voila, you're immune to like whatever. You have to like make it get into cells. You have to make sure the cell doesn't just immediately destroy it. You have to make sure it gets to the right cells. For mRNA vaccine, it doesn't really matter that much, but for CRISPR-Cas9, it does. You have to, if you wanted it to actually affect your musculature, you would have to get it into like at least a majority of your muscle cells, which is like a ridiculous undertaking. I don't know of any technology that does that. So yeah, there's just a lot of flaws. When you inject it into your arm, you're injecting into like interstitial space that's just going to diffuse and get degraded away, and it's not going to make it into any
like it's just not even close to the specific site it would need to be at.
I mean, you could inject it right into a muscle, and it might get into a couple muscle fibers.
So are a lot of these CRISPR home kits just probably not even doing anything?
Oh, they're not doing anything. Yeah,
like they're just literally like probably your body immediately fights it off. Boom, done.
It probably just degrades it because it's either I don't know what they're made out of. Either they're the actual protein and then a gRNA to target whatever gene.
Yeah.
And protein that's going to degrade pretty rapidly upon injection into the body, or it's like mRNA or something that will make your cells produce the Cas9 protein itself and hopefully tag up with the gRNA, but that can be degraded super rapidly as well. So like either way you look at it, it's like they're not going to have the sophisticated like delivery mechanism needed to get it into all of your muscle fibers. Like
how far away are we from actually having this stuff work? Do you think, like, how many years before? Because if people are trying it, do you think eventually they'll hit hit the bullseye on the dartboard, or is it just like
there's still a lot of like, people looking into delivery of the CRISPR-Cas9 system. So, like for example, with the mRNA, there's also like liposomal-based delivery methods to try to encode it in like a fatty bubble and get it into the cells. Other people are trying to do it with the adeno-associated viruses. They're these like they're basically these little viruses that can't replicate, but they're able to infect cells and then release their payload to allow for whatever. So people are doing that as well. You know, it's still all work in progress. So you know,
they succeeded with. They succeeded with mosquitoes, right? And the biggest thing, they made it mosquitoes so that they can breed once.
Yeah,
and then all their offspring can't breed anymore. They use that like Disney World or something.
Out of the George Church lab or something is really cool idea. Basically, that's called a gene drive, and they basically edited a mosquito and somewhere in gene. I don't know which gene they're targeting, but it basically makes them infertile, or it makes their progeny infertile. So it probably affects like the germline, but not like the whole mosquito, you know, so that their progeny can't reproduce. And so the idea is that these mosquitoes that, you know, carry this basically like species destroying mutation are like spreading out and like having babies everywhere, and then that whole generation of mosquitoes will be duds.
Because humanity's kind of agreed that the world's just better without mosquitoes. I've heard that before, right?
I think,
like
that's
if
kind
you
of
remove them from the
partly why
ecosystem, there's not a lot of consequences,
and
it just maybe eliminated a mass amount of human
right
disease.
I mean, that's what I've heard, and like that's why I think they're like allowed to do it. But they're a huge like disease vector, obviously. So there's a lot of benefits to it. I always feel like super suspicious though, because I'm like, you're knocking out an entire species.
It feels like there might be more things.
Like,
involved.
is there
It's
something
like a worldwide species? Like, are there secondary, tertiary effects that we have not considered for this?
I remember
very possible.
I
remember talking. I can't remember who it was, but I was talking to somebody who was getting in environmental sciences, and they basically go, Yeah, we've basically discovered that we suck at changing ecosystems. Like it's because the ecosystem is so much more complicated than we think. Like even the mosquito thing, we go, oh, we can knock these out, and all the birds and things that eat them, they can eat something else.
Right.
But then there's something we won't think of where they'll start eating some other bug we care about. There's all of a sudden there's none of that bug, which affects this thing. So it's like, the ecosystems are such a machine. It's like if you went up to a car and you're like, I don't think we need this carbonator anymore. So, but we don't know if we're pulling out we don't know what we're pulling out. We don't like how important are mosquitoes? Are they not important or not?
That's what
like
I'm saying, with like CRISPR-Cas9 gene editing, it's like we don't know enough like about biology in general. We can just go out knocking out some gene or something, and then hope nothing else bad happens.
It's like you can get these wildly specific results in a super controlled lab, but then when you go actually put it in the world, it's anything but controlled. And so it's like, what were you controlling for that's no longer controlled for that might affect the outcome? It's like, I just remember the Bill Nye video when I don't know if you guys saw this Bill Nye video where he had like the pyramid of all the blocks, and each block had like a different species on it. Like it was like a Jenga tower, and he was like showing like, you can pull out a few and maybe the tower won't fall over, but pull out the wrong one or pull out too many, the whole thing collapses.
Right.
It's always been in my brain.
Like, for sure.
Yeah.
I have a question for you. So if we start mixing crazy technologies, how familiar are you with deep learning, like the computational?
I'm a little familiar with AI stuff, and it's definitely not my field, but I'm like aware of it. Yeah.
I'm wondering deep learning's effect on medical research. And we're stepping out of so just to preface for everyone listening, and obviously they probably assume this anyway, we're stepping out of play. I know barely anything about this. So we're just in total skeptic land. This is not, we are stepping out of the viable, we're stepping out of the viable understood science that you could look up to like, we're just in playland. But deep learning, from my understanding, deep learning is basically it's similar to a human brain, and it just looks for patterns, and you don't have to tell it what patterns to look for. It just looks for them, and it self-fixes its own prediction errors. So if it looks like you just give it tons of pictures of dogs and cats, and every time that it misidentifies one, it fixes itself, like routes it back, so it won't make the mistake so much. Right. And so it can basically find these patterns at a rate and take in more data than a human or tons of humans can. So it can discover things that we just aren't capable of. And so I'm wondering, with medical research, how you could do that. You might be limited because you have to do simulations, and the world isn't a perfect simulation.
Well, actually, there are talks that I've been to or heard about where they talk about using different like AI algorithms and stuff for processing images, like pathology images and stuff like that, looking at tissue stains and stuff, and being able to do basically pattern recognition and try to either diagnose something or being able to create like these predictive they have like predictive algorithms for trying to like say like this person is this susceptible to this disease or something like that.
Oh.
Blah, blah, blah. So I've like heard about that stuff going on, and there's definitely a lot of research going on that as well, for being able, especially using them to look at like diagnostic images and stuff like that.
That's such a good point, where you could eliminate misdiagnoses, or at least make it really, really a small percentage of misdiagnoses.
Yeah, or at least like aid, right? Because it's always was it Bayesian theorem or Boolean? No, Bayesian theorem, where you're basically like you do a bunch of diagnostic tests, and like depending on the results of each of those tests, you can like at the end have like a percent like chance that this person has this disease or something like that. That's why like a doctor orders several different tests, because one, maybe you got scored positive on this one test, and it's like, okay, that means there's a 80% chance that you actually have this disease based on just how this test works. And so they pair that with another test and another test to like try to approach like a certainty that this person has it. So I could see it being used like that, using it as like another way of like, increasing the chance, increasing your confidence that this person has like a certain disease or something like that.
That would be insane. Yeah, you basically, because you just basically increase the percentage, like you're saying, the percentage chance that you know what the actual issue is. Instead of it being like 75, 80, now it's close to like 95, 90,
Right.
and you can, and yeah. I could see that because I remember when I used to watch House, that was a big problem. Medical dramas, they didn't know what was wrong with somebody till, an hour into the show. They misdiagnosed them, gave them the wrong medicine.
Yeah.
It's always lupus. It's always lupus in that show. Yeah, but I remember that being an issue. And I mean, obviously it's a TV show, but I feel like that would be an issue with our bodies are so complicated. How do you figure out exactly what the issue is if it's not just a broken arm? Especially when you have symptoms that are really similar to other diseases. It's hard. It's like, okay, well, what is this? So yeah, I wonder if it can find patterns that we're not aware of.
Yeah. Yeah, or make it easier, streamline it at least. Yeah.
Mm-hmm.
It might put a lot of pathologists out of work, though. It's not nearly that good yet, but
Yeah. Deep learning is going to put a lot of people out of work, man. It's going to be, I mean, in good and bad ways, but I think it's going to change our jobs, and our jobs are going to be, I think they're going to be a lot less technical and a lot more human interaction type jobs.
Yeah.
I mean, I think way in the future, it'll be more like entertainment jobs, sales jobs, because we're going to have so much information and everything's going to be automated. So all your base functions are going to be automated. They're going to build the buildings for you to live in. They'll farm the food for you to eat. They'll filter the water for you. All these machines will do that. And so at some point, it's like, what do you do with your time? It's going to be entertainment based, or, you're going to be a talk. Everyone's going to be a talk show host. Yeah, and art.
so
Everyone's
many
gonna
podcasts.
Everyone's going to have a podcast. But yeah, I'm excited for the deep learning world, man. I want to plug in. Me and Jack were talking about that when he was here. The Neuralink. I want to plug into the deep learning network. Upgrade, baby.
Yeah, just
start going full Skynet.
Yeah,
exactly.
Like triple, quadruple, 100 times your computing power.
Yeah, exactly. That's the future.
We'd have to watch out. If Wes had 100 times his computing power,
we're fucked.
We would be fucked.
Wes, he already has 100 times computing power from us. I can't imagine giving him 100 times that. It'd be game over.
100 times the computing power of like a Windows 98 or something.
Yeah, exactly. Sweet. Well, Wes, I really appreciate you coming on, man. This is really cool.
Yeah, this is a lot of fun. That was very informative, and I feel like there's a little knowledge gap that was cleared up a little bit.
Yeah,
at
thanks
least
for
me.
having me on. It's good to see
Yeah,
you
dude.
guys.
It's really enjoy it. Also, congrats. Are you married now, or did you guys have a small wedding, or did you do the elope?
Yeah.
Yeah, we eloped. Yeah, we went and eloped, but we're going to have the wedding in August, so that's like on the table. We still have to like send out invites and everything, but
everyone's gonna show up. You know what, though?
Congratulations.
Yeah, thank you.
Yeah, dude. This could be better because it'll just be a reception. That's the part of the wedding people actually want. You guys are already married, so we just go straight to the reception.
Just get right to the drinking. Yeah.
Yeah. You're basically just having a party. That's for sure. Yeah,
all
you gotta get
family and grandmas. Yeah.
You got to get all your furniture, your instant pots, your air fryers, all the kitchen gifts.
Oh,
yeah, you got to get all those. The
you
curtains.
Should start looking into some mRNA solution for hangovers.
Yeah.
They just in the morning I just give myself my little mRNA shot.
Yeah.
Stop the bad alcohol gene from doing its thing, and then boom, done.
Yeah, there we go.
Anyway,
there we go.
Just giving you some ideas,
I'll put it in the notebook.
Yeah.
That's anytime you need ideas for some more research, let us know. We got the ideas in here.
Mountain.
Yeah, the execution, we have absolutely no idea. That's what you're for, though.
Yeah. You got to start with something that people want, and then the execution stuff comes later. You figure it out.
Those are just logistics. Figuring out that's
logistics.
Yeah, we can figure that out. It's just exactly. What do the people want? That's what we can figure out.
Yep.
Well, Wes, thank you so much. If you are moving, if you're ever in Dallas, I'll be there next week. I got a 2-bedroom. You're always welcome.
Place to crash. Just in case.
Fun city if you want to go explore. That's why I got 2 bedrooms. I want people to visit. So, but yeah, it's good to see you, man. It's been a long time, and let's not go this long between
Yeah.
times. Next time.
Yeah. Let me know. Let me know. We're gonna go get drunk up in Pittsburgh. Let me know when
yeah.
that is.
yeah.
Date
Come
and
on down
time.
to
I'll
Pittsburgh.
be there.
We have a nice little house. We have three guest bedrooms if you want.
Ooh,
Wow.
There we go.
It's a house from the 1900s. There's no
Ooh.
heating
What?
up there, so it gets pretty cold. There's no
Ghosts.
cooling either.
Oh my god.
You know, if you want to pretend like you're sleeping under the stars, but not have stars to look at,
Then to come
I'm
through.
just like, yeah.
That man, I'm so glad you said that because I've been trying to find that. But everyone has AC now and
Hard to
heating.
come. I know it's
so
hard to get this nice vintage, air control. It's I
It's
don't
like
know if I've
camping.
been up there for like a year because I just have no reason to go up there. It's too cold or it's too hot. But
Wait, are you not in Pittsburgh? Are you not in Pittsburgh?
I'm in Pittsburg.
No,
Oh.
he's just got an upstairs of his house that he's
I
never
have
Oh.
walked into.
upstairs that I don't go to because it's too cold or too hot.
You haven't been there. There's someone living up there. You just have no
It could
idea. You have
be.
this roommate.
Someone might have moved in and I didn't even know it.
Oh man, dude. Well, it was awesome, Wes. You rock. Keep cranking. 3 years. Man, I just can't wait. You're gonna be a doctor pretty soon. We're gonna see you on the news. Wes Brown inventing mutated
You'll be
zebrafish that are taking over the
You'll
world.
be the next Fauci for the next disease. You'll be like,
Yeah, there we
telling
go.
everybody- Telling everybody
Yeah.
what's going on.
Everybody, stop
Get
injecting
2
yourselves
hours
with
of sleep a
CRISPR.
night sounds
Yeah.
great.
Yeah. You can come out,
right,
tell us to wear masks or not. That's what
Uh-huh.
we need.
2 masks.
All right, everyone that's
Great.
listening, thank you for tuning in, and we will see you next week.
Thanks everybody.
Keep podcasting. See ya.
Yeah.